All right, welcome back, fellow scientists. So in previous videos, we have looked at the history of DNA. Mr. Anderson from Bozeman Biology talked about the structure of DNA. Thank you very much. So today we are going to get into DNA replication. That's the big topic for today. That's the main topic. So go ahead and get out a new piece of paper. Move this real quick. All right, cool. So here is the big picture, right? Today we're going to look at how your cell takes one copy of DNA and makes a second identical copy. Okay, why does this need to happen? Well, you already know the answer to that. This needs to happen because of mitosis and meiosis. We have our parent cell right here. We go through all the phases and we get two daughter cells, and both of these daughter cells have to have this exact same DNA as the parent cell. Otherwise, they can't function. Okay, so that's why this has to happen. When does this have to happen? Well, you also already know the answer to that. This has to happen during S phase, right? So G1, S, G2, prophase, metaphase, anaphase, telophase, cytokinesis, those are all the interphase and then mitosis and cytokinesis of the cell cycle, right? So during S phase, we take one copy of DNA, we take our propeller-shaped chromosome, and then we duplicate it, we photocopy it, and we make it into an X-shaped chromosome. All right, so what are we talking about? DNA replication. Why does this have to happen? Because of mitosis and meiosis. When does this happen? It happens during S phase. How does this happen? That's the main thing that we're going to talk about in this video, is how DNA replication happens. So go ahead, maybe get out a new page, right? So we're going to do a before, during, and after. This might take up like a whole entire notebook page, so just kind of be ready for that. But it's in 3 sections, so yeah, I'll let you organize your notebook the way that you want to. So go ahead and write down before DNA replication. This is kind of how we're going to represent our DNA. Right, so obviously DNA is in a double helix, but obviously it's really hard to draw the bases into the double helix. So go ahead and draw this little picture right here, right? This is the double helix, but then we need the flat spot because we need to represent the bases with letters. So obviously DNA doesn't normally have that flat spot, but we're just drawing it. Just imagine a double helix, you know, all the way throughout. The line represents the sugar phosphate backbone that Mr. Anderson taught us about, and then of course the letters represent the bases. So here's a little challenge for you. Using Chargaff's rules, after you have this filled in, go ahead and fill in the bases on this other side of the double helix, on the other side of the twisted ladder, right? So go ahead, pause the video, do that. Okay, perfect. So welcome back. Hopefully you remembered that A pairs with T and that G pairs with C. So this is what DNA looks like before DNA replication. So now go ahead and write during DNA replication. And by the way, I realize I'm moving fast. It's a video. Pause it if you need to. This is really important stuff. So during DNA replication, we're going to draw a basic DNA structure that looks something like that. We already have our bases filled in, right? Well, Mr. Leo, there's a bigger bubble. It's no longer a flat spot. I know. Do you know why? Aha, I'll show you. Because of this little enzyme right here called DNA helicase. So go ahead and write that down, right? Draw this little circle right here that represents the enzyme. It ends in ase. We know it's an enzyme. Remember back in chapter 2, we talked about how ose, glucose, and lactose are sugars, and they end in ose. And ase are peroxidase that was found in chicken livers that breaks down hydrogen peroxide into water and oxygen, right? That ends in ase, and most enzymes end in ase. I won't say all because biology is frustrating that way. But DNA helicase follows that rule. Ase is an enzyme, and DNA helicase unwinds the DNA double helix. So we can see that. Here's our DNA double helix, and then this is what it actually looks like, right? When we're up here, like it doesn't actually have that flat spot. We kind of just do that for convenience. But this is what it actually looks like. This is called a replication bubble right here. Well, Mr. Leo, how in the world did this side? Well, it has another DNA helicase that's traveling that way, and then this DNA helicase that I already drew is traveling that way. Actually, that's opposite because of the video. Anyways, moving on. So DNA helicase, that's the first enzyme that's involved in DNA replication, and that unwinds the DNA double helix. And then the second enzyme, and I would argue probably the more important enzyme, is DNA polymerase. So write this down. Draw in this little blue circle. This is DNA polymerase, right? And DNA polymerase is going to travel this way. Okay, and DNA polymerase, what it does is it adds nucleotides to the 3' end of the growing strand. Remember, Mr. Anderson taught us about the 3' and 5' and how DNA is anti-parallel in its structure. We'll talk more about that in the next video. This is just a basic overview of DNA replication. We're going to get a little bit more in depth in the next video and see why DNA polymerase actually has to add nucleotides only to the 3' end, why it can't add them to the 5' end. But that's the next video. So go ahead and draw that and write down the definition. And then, of course, we have our DNA polymerase down here that is going this way, right? So what DNA polymerase does, there are free nucleotides just kind of floating around in the nucleus. Remember, because that's where DNA lives, so that's where this process happens. So there are free nucleotides floating around. So DNA polymerase will just say, oh, we have a G. Okay, well, C binds with G. And so it will add that nucleotide. There's the base. There's the part of the sugar phosphate backbone. And then it's going to move along, right? And then it's going to add a to a T and a T to an A, and then AA to the T, right? And then it's going to do the same thing. This DNA polymerase is going to do the same thing going this way. And so we have symbolized in red here the new strand, the growing new strand. And again, we'll talk about 3', 5' in the next video. So that's our picture of during DNA replication. And then after DNA replication, go ahead and write down after. All right, so this is kind of our structure. This is kind of our backbone of what it looks like. So we have our old backbone pictured in black and our new backbone pictured in red. And then let's see if we can find it. Aha, there we go. Perfect. So again, old backbone in black, new backbone in red. So our old backbone, right, would have the following bases, and we already know that because that's what it looked like before. And so the same bases are in the same places. That rhymes. I'm a poet and I didn't even know it. So now go ahead, pause the video. Make sure that you have your Chargaff base pairing rules firmly cemented in your head, and fill in the new strand, the bases that would be on the new strand. So go ahead, pause the video. All right, perfect. Welcome back. So hopefully you remembered again that A pairs with T and C pairs with G, and A pairs with T and C pairs with G. So on and so forth. And so that's what we get after DNA replication. Now, notice two things. Number one, we have 2 exact copies. So each daughter cell gets an exact copy of the DNA that the parent cell had. That's very, very important because we want our daughter cells to have the exact DNA, because otherwise they can't function. They wouldn't know what to do. There would be no boss of the cell. There would be no DNA to control the cell. Okay, so that's thing number one, 2 exact copies. Thing number 2, DNA replication is semi-conservative. So you'll notice that each of our double helixes has one old piece of backbone and one new piece of backbone, right? So that's what's known as semi-conservative. So the old DNA provides a template to make the new DNA, and because of Chargaff's base pairing rules, because of the way that God made it, now we get 2 exact copies, and it's very elegant and very simple, but yet complex at the same time, right? Praise the Lord. So this has been Mr. Leo. Today we have learned just the very basics of DNA replication. Thank you so much for watching, and I will see you on the flip side.